Welcome to this episode of Inspirational Women in Neuroscience. Today we will hear from Professor Dr. Inga Neumann. As a researcher and professor of neurobiology, she studies the role and function of oxytocin in the brain. We are very fortunate to have the opportunity to be joined by such an intelligent and quite honestly inspirational scientist. Enjoy hearing from Professor Dr. Inga Neumann.
To be talking with you today. I heard about you through the book Quirk, and the author described you as, and I'm pretty sure this is verbatim, a quote from the book, but it said, pretty chick for a scientist and nonetheless professional and insightful. And after I read that, I thought to myself, I have to talk to this neuroscientist. She seems awesome, and your work really seems incredible from what I've read. So thank you for taking the time to talk to me today.
Oh, you're very welcome. Of course. Don't speak so fast, so that would be easier for me.
No, yeah. Of course. So the research of yours that is highlighted in the book is about how oxytocin affects the brain and our behaviors. And I would love to come back and talk about your research, about this research that was in the book in a bit. But okay. I first want to know, how did your interest in this area of study start?
Yeah, you must know my little bit historical background. I'm from the GDR, from East Germany, and so I started neuroscience at a very, very technically extremely low level with a lot of creativity to build things which were not commercially available. So we were very creative, and what actually happened in that time, that we were in our lab, we developed the one of still the best radioimmunoassay for oxytocin and vasopressin at this time. And that allowed us to monitor not only oxytocin into blood, the blood concentrations, but also oxytocin released within the brain. And that was indeed at this time something extremely innovative, because at this time, that was in the 80s, so old I am already, at this time we knew there are fibers in the brain, somehow a few fibers, but nobody had an idea when these fibers are activated and what is oxytocin in the brain doing. So there was absolutely almost no knowledge, especially for oxytocin, because at this time vasopressin was much, much more in the focus of scientists because of its anxiogenic effects, and it was about targeting vasopressin can treat depression and anxiety disorders and things like that. And I was always a little bit like, oh, the poor Inga Neumann and with her oxytocin. Yeah, nobody is interested really in oxytocin at this time. And so I started studying intracerebral release of oxytocin in the brain. And I thought, okay, it's known, oxytocin, the word comes from the Greek word for quick birth. It was already known since 1906 when Sir Henry Dale discovered that pituitary extracts promoted and facilitated and speeded up delivery and uterine contractions. And I thought, if this has this function in the periphery promoting birth, what is this doing in the brain? Is this also released in the brain during birth? Or the same during lactation? Peripherally, it's known oxytocin is very essential for milk let down during the milk ejection reflex. And I thought, so maybe let's see if the brain is also, if there's also oxytocin released in the brain during lactation. So these were my very, very first studies, in fact, and oxytocin was found to be released in different parts of the brain. And later on, I became then interested really to study what is this centrally released, this endogenous oxytocin doing in the brain. And what fascinates me until now is that this is such a fantastic thing Mother Nature has invented, that you have a peptide like oxytocin, a small nonapeptide, which is released into the blood to fulfill or to support physiological functions like birth or like milk, like nutrition of the offspring. But at the very, very same time, oxytocin is also released within the brain, and there it enables that the mother really wants to take care of the offspring. It's not only that she can do it because there's birth and because there's milk, but that she's also motivated to care for her offspring. So that was my first start into neurobiology, actually, with oxytocin release in the brain, and these mechanisms are still fascinating. And then later on, we discovered that oxytocin is not only important for reproduction and maternal behavior, but for many, many other social behaviors, and also for emotionality and also for stress. So at the same time, if you take lactation, for example, you have a lactating brain. I mean, you have a mother animal or a mother human mother, and in lactation, the oxytocin system is highly activated. So which means the neurons in the brain, in the hypothalamus, they produce much more oxytocin. There are more oxytocin receptors in the brain, and we are not talking about the periphery, of course, that's also activated. And you have lots of release in the brain during suckling. And we found that this high activity, so the brain is full of oxytocin, and the maternal brain, the mothers, of course, so focused on the offspring and on the baby and so on. And that is also has important other side effects, because this high level of brain oxytocin activity is important for the reduced stress response the mother shows at this time in lactation. And reduced stress response means not only reduced pituitary release of ACTH and glucocorticoids from the adrenal glands, but this also means reduced anxiety-related behavior in animals, or in humans, let's say, reduced anxiety level and kind of sedation. So that is an extremely important aspect of the peripartum period. And then we thought, okay, so we could find that stress responsiveness is then really also reduced. And that was, in fact, these are studies which I performed at the Max Planck Institute in Munich at this time, when I was somehow infected with anxiety and stress research, and I thought, oh, this is cool. Let's link the nonapeptide research on oxytocin reproduction, also with stress and with emotionality, anxiety, stress coping strategies, and things like that. So this all came a little bit then together, and in studying females during reproduction, and I've also fascinated, also not only oxytocin, also prolactin, for example, another typical female reproductive hormone, which is important for milk synthesis. Also prolactin is synthesized in the brain, is released in the brain, and is also important for maternal behaviors, important for anxiety and reduced anxiety, especially in lactation. So this all comes nicely together, but then came an important thought, and I thought, this is all about female. This is such a fascinating effect only in females. Is there physiological justice? Is there also a situation in males when they can profit from the endogenous oxytocin system, right? And of course, if you think of males, you think of mating, right, and sexual activity. And that's when we started then mating and sexual behavior, and when we saw that during mating, oxytocin is also released in the brain, and it has exactly that anxiolytic, this anxiety-reducing effect as we see in the lactation period. So there is indeed justice with respect to the effects of oxytocin at some point.
Yeah, it's interesting because a lot of the studies surrounding oxytocin really look at mothers and that period with motherhood and having an infant. And I think it's really interesting that you looked at how oxytocin affects the male brain, because that's something that really doesn't have a ton of research on. But I also found it interesting that you had focused on how oxytocin plays a role in the autistic brain. And I was wondering if you could maybe explain your studies on face
Did you say
recognition.
autistic
Yes.
brain? Yes, okay.
And how oxytocin is contributing to how we recognize faces and recognize like this sort of social side of us. So I was wondering if you could explain what the role of oxytocin with someone who has autism.
Okay, so first of all, I'm a biologist. I'm a bloody biologist. I'm not a psychologist, so I'm not working with humans, except we do some small studies with saliva, oxytocin in saliva, or things like that, which we can do. But so that is so I did not perform studies on face recognition. What we know is, of course, and so all this work with oxytocin was really boosted, let's say, by the first human studies on oxytocin applied intranasally, and that started with a description that oxytocin increases trust behavior in humans, although this has never been replicated, I must admit. But many papers describe pro-social effects of oxytocin, which includes increased, let's say, face recognition in healthy volunteers. But there are also first studies describing that oxytocin in autistic patients, that this increases some aspects of social behavior, and that fits also with our experimental work on animals, because we can show that oxytocin in the brain, the endogenous oxytocin system, is essential for naturally occurring social preference behavior, which you can find in rats and mice, for example, and in both species we have studied that. So a mouse and a rat, if they are in their cage and that you introduce another conspecific in the cage, they would be extremely keen to explore the conspecific. In contrast, when you just place an object into the cage, they would not be so interested to explore the object. So that means social preference behavior. They would always prefer a social stimulus versus an object stimulus, and that behavior is extremely dependent on the brain oxytocin system. If you block the system, they don't show that. And in that line of research, and of course autism is also linked to aspects of social avoidance and social fear. So we know if you experimentally stress-induced, if you induce, let's say, social avoidance behavior, for example, by a social defeat exposure, then you can reinstate social preference behavior with oxytocin. And in another model we are currently working on, which is a model of social fear conditioning. So we have a mouse model where we can induce social fear in that way that the animals, mice in that case, they receive a very mild food shock while they are actively exploring a conspecific. So while they show this social preference behavior and they learn, oh, social exploration is very harmful, you get a food shock. It's a very mild food shock of 0.7 milliampere, so that is just that they realize that it's unpleasant, but it's not really harmful. And the next day, the animals would selectively avoid conspecifics, and that's what we call then social fear. So it's a wonderful model for social fear, and we know that oxytocin in the brain, specifically within the lateral septum, has amazing effects and very selective effect on this to reverse this social fear, whereas in contrast, non-social fear, in a non-social fear conditioning paradigm like acute fear conditioning, it may even impair fear extinction. So the oxytocin in the social contact has extremely strong positive effects to promote social interactions and to heal or to reverse social fear, for example. So, and therefore we are quite optimistic, and first results also from other groups in Australia and other places are showing that oxytocin applied intranasally may have beneficial effects also for autistic people. However, I'm always the one who is a little bit careful with such a general statement, because what we have to consider, and we are currently doing more research on that, because if you apply oxytocin nasally over weeks, right, daily, twice, 3 times daily, you have to consider, and there are studies also available from animal work, if you apply oxytocin chronically or repeatedly over a longer time, then you have dramatic effects on the endogenous oxytocin system. First of all, you have a downregulation of oxytocin receptors, and that is, of course, then what you do not want in autistic children. So I would not give my child, if this would be autistic, daily oxytocin intranasally, because you may significantly harm the endogenous oxytocin system. So that is that has to be considered, and we have to be very aware about that, because once a single treatment will not be long-lasting, of course.
Mm-hmm. And you were talking about how you focus on the biology of animals, because I mean, it's much easier to take an intensive look at the brain of mice and rodents when it's doing activity than, of course, with a human. So I'm wondering, how do the differences between rodents and humans affect your experiments, and how you can apply them to human behavior?
Yeah, that is a very interesting question, of course, always. And I mean, you have to go into brain neuroanatomy. I mean, the brain is, of course, in general, brain structures, or let's say neuronal structures or neurons, and the functioning of neurons is highly evolutionary conserved. At the same time, you have to say, of course, they are big species-dependent differences. If you think of cortical functions, especially, if you think of cognition and learning memory and so on. But from the neuroanatomical point of view, for example, various parts of the limbic system, and that is where oxytocin acts mostly, limbic system like the amygdala or the lateral septum, the medial septal nuclei, the nucleus accumbens, the hypothalamus, of course, the thalamus, those regions are highly conserved in evolution. So, and therefore also emotions like anxiety or stress coping, they are also, with respect to their regulation, highly conserved. There are the same neuropeptides in the brain, the same neurotransmitters in the brain. There is, of course, a varying degree of complexity, of course, in the brain, but in general, the limbic system works pretty much the same. Of course, if we are afraid of something, we do not necessarily run away. Yeah, but we will, because we have the cortex, we can say, okay, this is just a noise of a plane, and I can stay here safe. It's okay, right? An animal would just run and hide, right? So we have the cortical control about our emotions, but in general, basic mechanisms, like especially those basic emotions like essential anxiety, are very similarly regulated, and so there is good evidence that lots of the knowledge we gain in animals will be we can translate into humans, although we have to be aware of the fact that, of course, human behavior is influenced by many other let's say cortical structures.
And listening to you speak now and reading some of your studies, I find that a common theme in your research seems to be that our hormones control so much of our lives. Like, they dictate how stressed we are, how loving we are towards our children, the amount of trust we show towards others, and I guess the list goes on and on. For me, this brings up questions of free will and of a sort of biological determinism. And I'm wondering, as a scientist, what do you think about this? Like, how much control do we have over our behavior versus our hormones?
Okay, yeah, this is of course nice. I just gave a lecture yesterday, and we had this discussion about the free will, which is a very interesting topic, and I disagree with those neuroscientists which say we do not have a free will. All I decide, I mean, of course, I have, so let's say the free will discussion is, of course, a longer one, and I cannot do that on the phone, actually. But I will try to get my point over here. Of course, if in case I am a criminal person, right, and I cannot avoid, let's say, killing other people, right? So, and then it is, of course, this consequence that you say, yeah, it's my brain. I can't help. I cannot avoid this. This is not my free will to do that. My hormones drive me to kill people, right? Then I disagree, because it is my free will, for example, to say, oh, I realize those tendencies in myself. I go to a psychotherapy, and heal these intentions, for example. That's my free will. I can also have the free will to say, okay, I leave my knife at home now, always when I go walking or something like that. So we have a lot of decision possibilities, and all my brain is doing for me, if people say the brain is deciding for me, it's still me. The brain decides on the basis of my education, of course my genes, of course. I cannot do anything against my genes. But as we know, because of epigenetics, genes can be modulated. So if I'm trained as a child, as an adolescent, or as a young adult, if I get trained to alter my behavior, not talking about killing, talking about emotions. There are psychotherapy, for example. There you can educate yourself to control your emotions consciously. We have the cortex to control our behavior, right? And it's a kind of personality thing, of course, and personality is me. That is me. If I'm able to control that, or if I'm less able to control that. So if I have the for example, or take pedophile people, right? And we have in Regensburg here we have treatment options for people who realize I like children more than I should like, right? Then they can anonymously go to that treatment center, right? Before they do something bad, right? So that is an act of free will. You cannot do anything against these innate, let's say, drives or innate behavioral tendencies, let's say, right? But you can, as a human being, you can consciously control them. If you cannot do that, that is then a matter of personality. And then you should not blame your genes or your brain and your hormones. Then it is a matter if you are able to control your emotions or you control your behavior. It's still your free will.
I get that. Like we all have these genes and hormones that are predetermined for us, and we can't control it. But what we do have control over is how we respond to these things that we're born with, and how we take responsibility to deal with them.
And these hormones don't do their things by itself. I mean, there's so much cortical control about the limbic system. There's so much control. There's a cingulate gyrus, a prefrontal cortex, the orbitofrontal cortex, the temporal lobe, and so on. There are so many brain regions which have amazing complex interactions with the amygdala, with the septum, with the hippocampus, and so on. So and via these regions are our cortical control about our behavior, let's say, and we can use it or we cannot use it, of course, to consciously control our behavior. Of course, sometimes if you just arrive at your car and a policeman just put you a ticket on your car, and you can kill this man, of course, right? Or you can at least beat it, or you can shout at him, but you can also say, okay, I better go away and better be very not patient but very polite. And excuse me, and maybe then I have a chance even that he's not giving me the bill, or it's too late, and then you say, okay, I better go to the court or whatever, right? So you can control, although you feel your emotions inside, and that is especially true if you are a mother of children. Sometimes you can just beat them because they behave terrible or whatever, right? But of course, as an educated person, you will not do that. You will control yourself. So we have this ability to control ourselves if we know the rules, the morals, the ethics, the laws, and so on and so on. So that is what we can be educated to, and that is all what we think about the free will. So that's all what belongs in the topic of free will. Yeah.
No, thank you. And sorry to pose such a difficult question over the phone. I know it can take, I mean, more than just 10 minutes and maybe hours to tease out that.
Yeah, it's
question.
An interesting discussion.
Yeah.
Okay.
And you mentioned you brought that up at a conference yesterday, or that was a discussion.
No, this was, I have lectures here, student lecturers, and we were talking about the limbic system, in fact, and somebody asked a question about the free will, and so we discussed that. Yeah.
Okay, well, thank you. And I just have Do you have time for maybe a couple more questions? Is that all right?
Naja, 10 minutes, but not more, because somebody's
Okay,
waiting outside. Yeah.
Oh, okay. I just as one last question, what advice would you have for, I guess, aspiring scientists in the field of neuroscience? What advice would you give them?
So which level? PhD level?
Whichever you, yeah, any
Okay, so
level.
what I always, and I'm always asked for advices for this, or I always give the advices to the students if they want or not. So I would, if you are really keen in neuroscience, I mean, and if you want to promote your career, I always advise go to the best labs you can think of. Go to the labs which have the state-of-the-art methods. Also make use of the fantastic methods which are available these days. I mean, we started and we thought it is very amazing in the 90s. We used antisense oligonucleotides, and we thought really, wow, that's the end of any development we can think of. Now you have amazing techniques. I mean, epigenetic is a fascinating field. That is, for me, one of the biggest discoveries of the last 20 years, that we now know how the environment shapes genes and how in that way environmental factors can over long-lasting time shape our behavior, for example. That is just absolutely amazing. And of course, we have techniques like optogenetics or chemogenetics and CRISPR-R and those things, and that is really amazing. And the progress we can make in the field is fascinating. So the advice is really stay in the field you like. Don't switch fields if you really like a field. I would never, I mean, as you have realized now, I started in my diploma work with neuropeptides, with oxytocin vasopressin release in the brain, and I'm still working with oxytocin vasopressin, of course other neuropeptides as well, like neuropeptide S, CRH, and so on. But so I could never ever imagine really leaving now the field and work in something completely different. That would kill me. I would never do it, right? So if you like a topic, if you really like a field, stick in the field and just deeper insight. So that is, don't change fields if not essentially needed. Of course, if you sometimes have not the chance with a job and so on, then it is difficult. Yeah. And another advice for women, of course. Another advice for women, I think, and that is now, have to be a bit more careful now over the phone. In the lectures, I say that different. But I mean, I think what is important, balance your life. If you are fascinated by science, you can be fascinated by science, but also let yourself be fascinated by being a mother, for example. I would never have liked to be just a scientist, a woman in science, but without children, I think I would have missed something. And now my children are out of the house; they are adults. One is studying biology and is becoming interested in neurobiology. I cannot imagine. I wonder how this comes. And but looking back, I think one of the things I'm really proud of, and that I was able to do both, to have children and to go forward in my scientific ways.
Well, thank you so much for talking today and giving some wonderful advice and sharing a bit about your work and experiences in the field of neuroscience. I think it's going to be great for people listening to hear from you, and I'm very, very happy and grateful that you were able to take the time to do this. So thank you so much.
You're welcome.
That concludes this episode of Inspirational Women in Neuroscience. A big thank you to Professor Dr. Neumann for joining us today.